Sure. Well, thank you all for being here. Thank you so much for the introduction. As was mentioned, I'm David Carlson. I'm going to be talking today about machine learning methods to learn improved EEG biomarkers in clinical trials. I'm a professor at Duke University, where I'm joined between the engineering school and the medical center. So I get to do a lot of interesting things between the two of them. And before I dive in, I wanted to start out by telling a joke that I was told when I started graduate school, which wasn't that long ago, which is, what is the difference between machine learning and artificial intelligence? Does anyone know? So the answer back then was that one works and the other never did, right? And it was all just a question of branding, right? That machine learning was this new thing, and it was a new thing, and we would never call it AI, because AI at that point in time was a pretty dirty word that had just gone through one of these AI limits. So I find this hilarious, thinking back on it, that we all started calling what we're doing now artificial intelligence, which it really is. But anyway, what we want to talk about today is trying to say, can we learn interpretable neural biomarkers? And can we find these biomarkers that are going to look at things like clinical outcomes, or conditions, or risk? And often we're going to do this in the context of neuropsychiatric disorders, so things like depression, or ADHD, or a lot of other things like that. But there's this question, there's this comment I want to start out with, that neural activity is really complex. I think everyone understands that the brain is really complex, does a lot of things all at once. I'm talking right now, I'm moving, I have emotions, there's a lot of things going on. I think it's pretty obvious that we need to figure out ways of doing this well. So the question is, well, if we have really complex data that we're measuring out of the brain, can we actually break the complex signals into interpretable patterns that are actually useful for doing things like prognosis and predictions? And can we relate these signals to susceptibility or treatment? And this last one, which we're going to talk about, can we be used to develop novel treatments? So if we understand more about the brain, can we actually use that fact to actually design treatments and actually change treatment paradigms? And so I'm going to talk about this in two contexts. I'm going to start with animal studies, which is where I kind of started this line of work. And I'm going to talk in particular about how we're trying to adapt it to clinical trials. So we're going to start by looking at what we can do in a mouse model. And so really, can actual machine learning technique help understand neural circuits? So if we're looking at a mouse, we want to understand, say, what's going on in this mouse's brain. And so this is an ongoing collaboration with Kafui Daraza, who's over there. He leads the Laboratory for Psychiatric Neuroengineering. And what we really want to do is understand how the brain acts and changes in animal models with neuropsychiatric disorders. And so we're trying to find early biomarkers here, and can we find early biomarkers that are encoded just in the neural activity? And so a lot of this motivation comes from trying to understand depression. So major depressive disorder there's a lot of things that go on with depression, and you have to have some amount of these in order to get the clinical diagnosis. But what's really interesting in depression, as of 2017, was labeled the most debilitating illness in the world by the World Health Organization. And so we wanted to figure out a way of trying to say, can we actually take depression, and rather than waiting until we know it's there to figure out ways of treating it, can we try to do it earlier? And this motivation actually comes from looking at heart disease. So if we look at heart disease, what happens is over time, you get older and older, and some people, when they're older, have a very normal heart function, whereas others actually have a heart attack or heart failure. And if you have a heart attack or heart failure, you can come up with ways of treating it. You can put in a pacemaker, for example. And these techniques are now well established. But a much better idea is to try to figure out who's going to go into which group much earlier. And so it was actually not that long ago, it's been many decades now, but it wasn't that long ago that we discovered that blood pressure and cholesterol was actually a very good biomarker of which group you were going to go in later on. And if you know that blood pressure and cholesterol is actually a very good biomarker of this, you can come up with a way to start treating it much earlier in life, and this has actually greatly decreased rates of heart attack and heart failure. And so the question is, can we actually do the same thing in depression? So we know that if you undergo severe stress, some people are fine after severely stressful situations. These are labeled as resilient in the literature. And some develop major depressive disorder. And these are going to be labeled as susceptible in the literature. And you have ways of trying to treat them after the fact. But there's this question of, can we actually do the same thing that we're doing when we are looking at things like heart disease? Can we actually try to figure out signals that will tell us what is going on? And so there's this question of, can we find some sort of signal that indicates vulnerability? Can we actually go and do this? And there's a couple things we know that do this right now, but there's this question of, can we actually use neural biomarkers here? So can we just take measurements of your brain and actually find biomarkers that are going to signal vulnerability? And if we do this, we're going to try to figure out ways of trying to treat these types of disorders before they really start manifesting themselves. And so we want to do this in an animal model, and we kind of have an issue, which is that this is our animal. Sometimes it's happy, it's sad, but generally if you look at an animal, you can't really ask it to go fill out a survey about how it's feeling today. I mean, you can, it's just not going to do very much. And so, well, this seems like it's a pretty big issue, but there's actually been a lot of recent work which has kind of started to view emotions as latent networks in the brain. So this was a study from another lab, but what they were doing was they were basically taking fMRI images, and then they were asking people to self-assess whether they were feeling content, amusement, surprise, fear, or anger. And there's different patterns of activation all over the brain. So there are different brain regions that actually get activated and used when you're feeling these different types of emotions. And so you can actually take just the measured brain activity from this fMRI and actually use it to predict what type of emotion a person is feeling right now. And you can do surprisingly well at this. An fMRI is actually a fairly slow measurement that takes a while to get good data, so it's actually surprising you can do this well. So the question was, can we actually take this idea of there being networks of brain activity that are signaling different types of emotional states and actually say, can we find a brain network that's going to signal susceptibility? And so to do this, we're going to look at this mouse model. And in this mouse model, you can get some really clean data. This is going to be much cleaner data than we can get in the corresponding human studies that I'm going to talk about later. And so you actually take this mouse, and it gets, there are some very trained mouse neurosurgeons that implant this head stage there. And what's actually happening is that there's a lot of wires that go into very specific regions in the brain. So you actually are able to put electrodes, and they're able to get into the exact same brain region every single time. And when you do this, you get two different types of signals. The top type of signal here is something called action potentials. And the idea here is that every single row corresponds to a single neuron, and it just has a little flash here when that individual neuron fires. And this is a typical type of data that people use when they're doing implanted electrodes. And we actually made an interesting choice, which was we weren't going to use these neural action potentials. We're actually going to focus on the low frequency oscillations. So these are your brain waves. They're the same things you can measure on the outside of your skull. So if you ever do like an EEG skull cap, this is the same type of signal you can get, but it's a much higher quality. And I'm not going to go into too much of this, but this is actually a lower quality of information than what you can get out of neural action potential, so it's a little bit surprising that we're doing this if you know anything about this area. But what actually happens here is that the neural oscillations here are actually conserved across individuals, whereas it's very hard to tell you if I'm measuring this neuron in person 1, are we measuring the same neuron in person 2? And so while these are much more precise information, we're going to use this information that's actually going to be consistent over different individuals. And so what actually happens in these local field potentials is they're really just these low frequency oscillations. They're just brain waves, and so you get your different brain regions here, and you can break it down and look into it at different frequency patterns. You can look at it in your delta, your theta, your beta, your gamma, and what's actually happening here is that you can say, well, you can see, if you look closely, you can see that there's actually a lot of correlation between these different brain regions. Some of them are more correlated than other, but the correlation is actually interesting in this context. So correlation is really coordination. So if your brain is coordinating over two different regions, this means that it is communicating in some way, either through direct connections of your neurons or indirect through other regions. But you know that these two brain regions are acting in a coordinate way. And so if we have this, and this is the type of signal we're looking at, can we actually take this, what looks to be a very complex signal, and actually learn networks of brain activity related to this task that we care about? And so the idea we came up with was we are going to build something called an electrical connectome. And this looks extremely complicated. I'm not going to lie, it's actually pretty complicated. But I want to explain what's going on here. So what's actually happening here is we have in each of these spokes out here, we have a different brain region. So we have our hippocampus, VTA, CEA, infralimbic cortex, etc. And what we have here is, you can see here that there is power in the 5 hertz band and power in the 15 hertz band. And these types of lines here are showing when there's a lot of statistical correlation in the power between these different brain regions. So this is a sign that these things are actually communicating. And so really what we're saying here is we found an electrical network, and it's at 5 hertz and 15 hertz, and it's pretty much connected across the whole brain. And the other thing you can do is you can actually try to find what regions lead and which regions lag. This is the same type of things you would do in Granger causality. So if you're looking at Granger causality, you have one time series leads another. You can do the exact same thing and try to figure out which brain regions are leading the other. And so there's this question here of, well, we have a very complicated network here, and can we actually find a network that looks like this that's going to give us this biomarker that we want? Is there a question?
Yes. Why are we distinguishing between the 2 to 8 hertz and the 12 to 20 hertz? And also, I've been told that Granger causality is a bad model for how the brain leads. So, could you elaborate?
Yeah. So what's actually happening here is that it's learning 2 different frequency bands. So it's learning one frequency band that's 2 to 8 hertz, and it's learning another frequency band that's 12 to 20 hertz. So it's just saying this network is operating on 2 different frequencies. That's really what's happening here. I'll get into some of those details in a bit. The other great question you were asking was about Granger causality being a poor model of the brain. It definitely is. There are cases where it's known to hold up, most notably memory formation. So actually between the hippocampus and thalamus it holds up pretty well. But it's definitely not holding up across this whole brain. But really you can think about this as we're capturing the statistical correlations there, and those statistical correlations are going to help us find better networks rather than real causal patterns.
Thank you.
Yep. So there's this idea that if we have this network, can we actually find the network that's related to our stress susceptibility? And well, we could go about doing this, but it's actually pretty hard. I think I started out by saying that these neural signals are really complex. So it's going to be very hard to have one network that describes all of our neural activity. So instead of saying we only have one network, what we're going to do is we're going to say we have an observational model that's going to have our LFP data at any given point in time. So we're just going to window it and say we can take 5 seconds of local field potential data, and we're going to model our observation here as a superposition of different electromes, and they're going to have different weights. So this is just a standard factor analysis model. So the idea here is that you have your first electron, it's weighted by this score right there that's specific to this window. You have your second electron that's weighted by the specific score that's specific to this window, and your third score, and so on. And so the idea is that at any given point in time, we can take our neural activity, and by mapping it through this latent function factor model, we can actually say, well, what actually represents our window is that our networks are constant through time, but we're allowing the strengths of these networks to vary. And this is the idea that sometimes you're happy. Your happy network should be pretty high. Sometimes you're sad. When you're sad, your sad network should be high. And these networks should actually be time evolving and dynamic because you don't do the same thing at every point in time. There's a lot of math that I'm hiding in here. And I'm going to hide it because I want to have time to go through and show you what we do when we adapt this to human studies, which actually will turn out to be quite a bit simpler. But this is a general idea of what we're doing. And so the question is...
Can you just explain these radial diagrams? I mean, the numeric, you know, the fact that I mean, I can understand, okay, a connection between nucleus accumbens and basolateral or anterior amygdala, but what is the relative thickness and location here?
There are two things. So one, the location here, it goes from 10, 20, 30, 40, 50. That's hertz. So that's what frequency it's oscillating on, so what the oscillation frequency of your pattern is. The relative strength, so the thicker lines mean there's more power in that band, and the thicker the connection is, it's just how strong that correlation is. So things that are really highly weighted for a strong correlation. So there's this question of, if we have this, can we actually take this and apply this to a mouse model of depression? So just to really quickly go through a result of what we can do in an animal model, we have 44 mice used in a behavioral study, and we have home cage. This is where the mice live. It's supposed to be very calming, relatively so. We have a novel environment. We have an empty forced interaction test, which just basically means they're in a novel environment. So it's slightly stressful because you just pick them up and move them. And then you have a forced interaction test where you actually take this mouse and they interact with a large threatening mouse, and so they're very stressed. And so you have three different levels of stress. And what's going to happen is we're going to run this twice, but we're going to have two groups of mice. Some of the mice are non-control mice that go through a chronic stress paradigm. This chronic stress paradigm is designed to figure out which of these mice are actually susceptible to depression or not. And then we have control mice that don't go through anything in between. They should be the same before and after. We're going to learn 20 features, and then we're going to try to predict whether we can get behavioral conditions, pre- and post-stress conditions, and stress resiliency. And so if we actually do this, what's going on here is we measure all our local field potentials. We have three different behavioral conditions. Home cage, they're alone. In the FIT empty, they're just in a new environment. They're kind of constrained, but it's likely stressful. In the CD1, this is a larger kind of aggressive mouse. It's designed to stress them out very much. There's a barrier so they cannot actually be hurt, but it probably does not feel that way. And what actually happens is that over time, in between these two different behavioral paradigms, there's a chronic social defeat stress, which generally a happy mouse is a social mouse. So what this is it's looking at the ratio between how much a mouse is choosing to be social versus non-social. And in the control group that doesn't go through anything, almost all the mice choose to be social. And then the chronically stressed mice, some of the mice are resilient and some of them are not. Some of them are susceptible, or they really have moved into a non-social paradigm. And so there's the question, if we have actually used this type of technique, does this actually hold up? And so the answer is yes. In fact, if we take an electome, this is an electome learned from the data. This is one of the 20 we have. And we can talk through the neural properties, but I think for this audience, the neural properties don't matter as much. What probably really matters is how strong this network is actually expressed. So what we're showing over here on the right is we're showing the average scores of three different groups: our non-stressed mice, our resilient mice, and our susceptible mice. And what actually happens here is we're averaging over all these groups. This is prior to any chronic stress. So in the home cage, this factor network score is pretty low, but it's higher in the susceptible. In our mildly stressful situation, it goes up. In our very stressful situation, it's also elevated. And half of our mice actually ended up being susceptible, and the half were resilient. So the fact that if you average the resilient and the susceptible together, you get the non-stress controls, that's actually exactly what you would expect. But what actually happens is that the susceptible goes up drastically after you go through this stress paradigm. But what's really interesting here is even if we take this pre-chronic stress event, where we actually say we have no idea who's actually going to respond poorly to the stress or not, the network score here, it actually turns out is incredibly strongly predictive of who is susceptible to stress. So we can actually tell in these animal models who is susceptible to stress completely. And you can go through and calculate p-values on this on some held-out mice, and it's really strong statistically. And so we were really excited about this. This seems a little bit far away from the clinic, but nobody has ever shown that you can do anything like this from neural signals alone to get these precursors of what's going on. And so this was a really exciting result. And so if you go and talk to any of the biologists about this and you show them this curve, they tend to ask you one question, which is this one. Is this real? Right? That's a real question. Because we just went through, we used a complicated machine learning approach that I did not explain the mathematical details of, and you find these really complex patterns, and you say, this network, which defines some electrical patterns that we can measure in your brain, defines exactly your susceptibility to things like an animal model of depression. So is this real? That's a very fundamental question. And so the answer appears to be yes, because we've actually tested this pattern across a wide number of additional behavioral paradigms now, or not even behavioral, so you can do a behavioral model of vulnerability. So there's a mouse model of childhood trauma, which I won't go into, but that actually spikes up this network as well, makes all the mice more susceptible. You have a physiological model of vulnerability, so interferon alpha, and a molecular model of vulnerability, so you can actually knock out some genes that are known to increase the risk of depression, and they also spike up this network. You can look at genetic risk factors, and they also spike up this network. And so what's shocking is that these electrical patterns that we are finding are actually holding up across a wide number of conditions. And this is really kind of a remarkable result that we were really excited about. And so this is really cool. We can now figure out with really actually strong biomarkers in an animal model what our signals of susceptibility are. And so we're continuing to work on these animal models, but the fact that this is a precision medicine workshop, I want to focus on a separate question, which is we have these really kind of shocking results in animal models. And in animal models, we can do a lot of things. We can actually do these interventions. We're not doing a chronic social defeat stress in humans. That's not a very ethical experiment. And so there's this question of, well, if we're not doing the exact same thing, can we actually take some of these techniques and some of these ideas and actually take them and apply them to human studies and clinical trials instead? And actually take these same ideas that we're getting really great results on in animal models and say, can we use these directly in clinical trials? And so in order to actually do this, we started talking with Jerry Dawson, who leads the Duke Center for Autism and Brain Development. And they've been starting to collect a lot of EEG data while they're looking at studies, both observational studies and also clinical treatments on children diagnosed with autism spectrum disorder. So they're starting to build up a very large cohort of children that are undergoing various treatments. And also, there's a lot of observational studies where they're just trying to understand what's going on. And it's now standard practice that while you do a lot of these things, you're actually putting EEG on in a number of the experiments in order to measure their neural activity while you do all these different things. And so there's this question of, well, can we actually take those same ideas I talked about and make them work in human data? And can we improve our understanding of the brain and its responses in clinical trials? And this is really exciting because they're actually building a very large set of data because they actually recently got awarded an NIH Autism Center of Excellence that's being designed to actually look at the intersection between autism spectrum disorder and ADHD and treatments for them. And so, well, can we do this? So we don't have very large data from these historical experiments, but in order to figure out if we could really just get the methods to actually port to working in humans rather than working in animals, we started working on a recently collected clinical trial. So there's a clinical trial that was completed about 2 years ago now. It had 25 2 to 7-year-old children, and they were actually given a single infusion of autologous umbilical cord blood, and I cannot pronounce that very well. But this is basically a stem cell treatment. So this is a novel stem cell treatment for children diagnosed with autism spectrum disorder. If anyone pays attention to this disease, this particular clinical trial is actually a very high-profile clinical trial that was on CNN and all sorts of things like that. And so this is a much smaller cohort than you would typically try to do with a machine learning method. But we wanted to try to answer two questions. One is, can we track EEG changes that happen after treatment? So can we actually track how your neural signals are changing as you go through treatment? And can we predict who will or will not show changes after treatment? So this is that predictive medicine question. Can we predict how much you're going to improve from a specific treatment? So the first thing we did, which I'm going to talk about, is we adapted that complex network strategy to work here. And I built a computational framework to address what I like to call little big data, which is a name that I think infuriates a lot of statisticians, but I think they all remember it, so that's okay. And I'll get into what that oxymoron means in a little bit. And if we can do it here, it's going to help us when we get these really much larger clinical trials that are currently ongoing. And I'm really excited about that. So the question is, can we actually do this? And, well, the first thing we need to do is say, well, we were working with local field potentials before. That's shown on the top. So this is 4 different brain regions, the local field potential, and you can see that there's a lot of things going on, and here you have a lot of squiggly lines. You can argue about whether you can make sense of those squiggly lines. This is the EEG signal. So the big difference here, these LFPs are measured inside the brain, the EEG measured on the top of your skull. It's a completely non-invasive measurement. And so the detail of information you get when you're looking at EEG, rather when you're looking at LFP, actually decreases greatly. You lose a lot of information by not doing brain surgery on people. But on humans, we are certainly not going to do a lot of brain surgeries to try to get higher quality information. Right? We're going to deal with non-invasive measurements. And so, well, if we do this, we want to keep some of the same approaches we were doing before, but we don't think we can learn a very complex network strategy that requires this high detail of information. So what we're going to do instead is we're going to develop a custom convolutional neural network approach that we called SyncNet. And I'm going to talk through what this convolutional neural network approach is, but we're going to do one other thing too, which is we're going to build a Gaussian process method as a pre-processing step that's actually built into our neural network. This step here does really one great thing for us, which is that it will allow us to actually use the exact same convolutional neural network on any different electrode layout that you can use. So it's not very standardized what type of device you use to collect across different labs, so we want to map them together. So I'm going to come back to that if we have time. I want to talk about this convolutional neural network. I'm going to claim that we have a custom convolutional neural network that's going to work for this small data cohort, and also is going to be interpretable. Now, if you've heard anything about convolutional neural networks, you should probably have alarms blaring in your head right now. I just told you we have a very small cohort of data, and typically when you look at CNNs, they're applied to very large data sets. The most famous result in a convolutional neural network is really on these big image corpus, which started with 1 million images, and I think companies now train them on over 1 billion images. And if you start looking at the numbers, 25 is actually a little bit away from 1 billion images. And so can we actually do this on different clinical trials? So there's one thing that's going to help us, which is that one of the things you typically do is you pretend you have a lot more measurements from an individual than you actually do. Because what you're going to do is you're going to window the data into smaller time chunks and consider them as a somewhat independent measurement. And we'll get back to how to correct that assumption in a little bit. But we're going to focus on what this convolutional neural network is doing. So there's this question of, well, can we actually use a convolutional neural network here? And can we interpret this convolutional neural network? So the big thing about this is it's a convolutional neural network, but it's the shallowest type of convolutional neural network you could possibly use, where there is only one convolutional layer. We're only going to use one convolutional layer. So we're going to take our input EEG data here. We're going to send it to our first filter bank. And that first filter bank is going to operate on this, and we'll talk about exactly what it's doing. But what we're going to do is from the output of this first filter bank, we're going to do a max pool step, which basically says of the output from this first filter bank, we only take the maximum value that comes out of this. And that reduces this to a single feature. We have a single scalar feature that comes from our first filter bank, and then we have a second scalar feature that comes from our second filter bank. and so on. So what we're doing here is if we have our input EEG data here, we have this single convolutional layer, we have k different filter banks. If we go through this, what we're really doing is we're just mapping this to a k-dimensional feature vector. K is going to be pretty small because we have a pretty small set of data, but that's our big goal here. Is we're going to take our input data and map it to a k-dimensional vector. So if we're doing that, we can actually view this. Instead of viewing this as a convolutional neural network, which can be very hard to interpret and understand what's going on, we can actually view this as a nonlinear feature extraction step. So we have view it like this. So we have our EEG data, we take a window of EEG data, we have a nonlinear feature extraction, we have a low-dimensional extracted feature, so this is just our k-dimensional vector, and then we're just going to do logistic regression. So there's a big argument that if we have logistic regression, I think most people would agree that logistic regression is a very interpretable model. If the regression weight is high, we think that there's a positive response to that outcome. If the regression weight is low, there's a negative correlation between this and the output. So we think that we can interpret our logistic regression weights. And I don't think that's controversial. So what's interesting then is, well, if we can interpret the logistic regression weights, can we actually interpret our nonlinear feature extraction here? So can we understand and interpret these extracted features? So that's the big question we have here. So if we want to make this convolutional neural network that we can understand and interpret, all we really have to do is understand this step. So that makes it sound really simple, and I will hopefully convince you that it actually is not so bad. And so how do we actually go and create a convolutional layer, a single convolutional layer, that I would argue that we can understand and interpret? And so what we're going to do is we're going to say, well, we're looking at EEG data, we know they're brainwaves. What's really important in brainwaves is frequency properties. So one of the most standard techniques you use to evaluate EEG is to actually look at power in a particular frequency band. And one of the ways you would often go about doing this is by either using a short-time Fourier transform or by using a wavelet transform. And we're going to say, well, if we think that a wavelet transform is a really useful thing to do, why are we trying to learn a generic convolutional filter? If we think they should look like wavelets, let's just constrain them to look like wavelets. So what we're going to do is for that first filter bank, we're going to say for the kth feature, this could just be 1, this could be our first feature. On each channel C, we're going to say that a convolutional filter looks like this. So we have a channel-specific weight. What this will allow you to do is that in your convolutional filter, it can say how much should we use this electrode versus this electrode versus this electrode. And then we're going to have the center of the frequency band. That's going to be the same across all channels in this feature. This beta here just comes from that Morley wavelet, and it's omega and beta here are going to control your frequency properties. And we will know exactly what frequency band we're looking at because they're just parameterized to look like a wavelet. So really, this just allows us to vary amplitude. And then we have a weird term here that can throw people off, that this is just a phase offset. So this will allow us to look at some neural synchrony. So if you have a signal in channel 1 and a signal in channel 2, you can actually say, well, there would be a particular type of information if they were completely opposite to one another. That might be some type of pattern we're looking for. So it's really just looking for phase offsets. So if we do this, there's a couple things. One, I would argue that we can actually interpret exactly what this filter bank is doing. It's mostly looking at a particular frequency band and then looking at the different power over all your different brain regions that you could look at. And it still allows more flexibility than a traditional approach. But what you can do here is you can actually say, well, if we have this, we can learn all these parameters. So we can learn our weights, we can learn our frequencies, we can learn our phase offsets, and we can learn of the beta there that's controlling the width of your frequency band. And it turns out that this is actually not so bad. And we're going to drastically reduce the number of parameters we have to learn compared to a classical CNN, where you completely set everything free. And so these frequency properties are well understood, so we just borrow that knowledge. So if we have a filter bank like this, we can actually visualize what it's doing. So real quickly, if you're going to extract these features, really what happens if you have this type of convolutional neural network, you take the summation of your channel here of this is in window T, channel C, this is your EEG signal, and you convolve it with that particular frequency, that particular filter bank only on that channel, and then we just take the maximum value that comes out of this vector. I don't want to spend too long here. But what I want to do instead is say, if we have this procedure, can we actually go through, apply this to our particular case that we cared about, and actually understand what it's using to make decisions? So what we're doing is we want to learn neurodynamics that change post-treatment. I was talking about trying to apply to this clinical trial. We have this clinical trial. And so what we're going to do is say, well, if we want to actually track the neural changes over time, we're going to use a surrogate task where we're going to look at baseline. So this is pre-treatment, 6 months post-treatment, and 12 months post-treatment. And we're going to try to predict exactly what treatment stage we're going to be looking at. So this is actually a 3-class classification rather than a 2-class classification. So we're going to try to predict which one of these 3 are. And we're going to try to apply this. And what actually happens is that if you go through, you can actually apply this convolutional neural net to this structure. And what can happen here is you have this brain here on the left. You have your frequency pattern here. So because it's parameterized to look like a wavelet, we can actually calculate exactly what frequency band we're looking at. So this particular filter bank said it's roughly about 30 hertz. This is about the spread of the feature we're looking at. You have some side lobes here. That's just because we have a discrete approximation on a wavelet, so we're actually showing you what the actual frequency spectrum is, and that just comes from the discrete approximation. And this is a visualization of what was going on in that B parameter that was controlling where you were looking at. So this is relative power, and it's saying that it uses the electrodes in this part of your brain really well. It uses the electrodes over here really well, and pretty much everything else it kind of ignores in this particular feature. It uses them very minimally. The B is not exactly 0, but we can tell you where it's looking on this particular filter. And then we say, well, can we actually understand what this particular feature means in terms of our classification? So what we can do is we can take an example from the baseline measurement, push it through this convolutional filter, and say, what is the actual score of that feature that came out from this? And so if we look at what happens at baseline, you get pretty high feature scores, and at 6 months and 12 months, they're much lower. So this is a feature that actually significantly reduces after this treatment. We don't have controls here yet, because this is a phase 1 clinical trial. So all I can say is that it does decrease after treatment. I can't say because of treatment. But this is really exciting. So this is a feature that I think we can interpret. It's actually much more expressive than what people traditionally do. And we can actually say exactly how this relates to our different treatment stages. And so this is the big idea. So we think we have an interpretable convolutional neural network. So the question is, if we have this interpretable convolutional neural network, does this actually help us? Is this something that actually we care about? And the answer is going to be yes, which actually kind of surprised me, I will be honest, because this is actually one of the simplest things I've ever done. You can argue about whether it's actually simple, but compared to my normal 50-layer convolutional neural net things I do, this doesn't seem so bad. So what we did is we evaluated by leave one participant out cross-validation. So we leave a complete individual out because we're trying to estimate how well you generalize to a new population. We're going to average our ability to predict over these different EEG windows. And we're trying to predict these 3 classes, baseline 6 months post-treatment or 12 months post-treatment. And so one of the most classical ways people go about doing this is actually calculating power spectral density features. So you basically filter to a particular frequency band, calculate the power in this particular filter frequency band, and see how well you actually do. And so here, if you randomly guess it's 3 classes, you get 1/3. If you just guess a dominant class, we have some dropout here where we did either miss a 6-month or a 12-month measurement for some individuals. So if we have that and we just guess the dominant class, which is baseline, we get 41% accuracy. So if you do these traditional approaches where you actually do power spectral density, and you might even combine it with a nice support vector machine, so you can do a kernel SVM here to get pretty good results, you actually do decently well. You get significantly above random guessing or baseline. You actually do get a lot of information out of these traditional approaches. But if you actually go and use a custom CNN-based approach, this is a custom CNN approach for EEG in the literature that somebody else developed, and this is the one we developed. You actually can get pretty large gains here. This is actually a pretty large gain in this type of data. We went from 41% just guessing dominant class to about 50% using what they're currently doing to about 60% accuracy now. And I will tell you the difference between these, look, there's uncertainty, we don't have that big a cohort. Are we doing better? I would argue slightly, but there's uncertainty there. I can't say with confidence that we're actually much better than this other convolutional neural network. I can say with confidence we are a lot better than the baseline approaches. But what I would argue is a completely interpretable framework. You can at least understand somewhat of what's going on in our network. And so this is kind of something that we were really, really excited about, and we wanted to keep working on. And real quick, David, when should I end? Because I know we got started a little late.
Ending at 12:30 is probably a good idea.
Okay. Well then, I will get through this really quickly, which is saying windowing data makes it seem much bigger than it really is. I told you we have 23 patients, but if we split up into these different windows of EEG data, like you typically do, you end up with 540 labeled samples because you do a lot of measurements at baseline, a lot of measurements at 6 months, a lot of measurements at 12 months. So if you just take all your labeled samples per patient, and you multiply it by the number of participants, we should always be saying participants. These are participants, not patients. So 22 times 540, you're passing over 10,000 labeled samples into your convolutional neural network. So if you're the convolutional neural network, this actually seems like pretty big data. And you're pretty happy. But if you think about this, that's probably not realistic. We don't really have 10,000 independent samples. So can we come up with ways of actually saying in a convolutional neural network that we actually have this type of measurement structure? And this is what I like to call little big data, where it seems like you have big data because you get a lot of longitudinal measurements from an individual participant, but really our N is very small because we only have a few participants. And so this is what I mean here. And so what we actually have is we're not trying to generalize from 10,000 measurements, we're trying to generalize from only a few participants to a new participant. And so, can we actually do this? And so, this turns out to be really important in this type of measurement domain. So if you were to actually take this type of data and look at it, I think it shouldn't come to a surprise to this audience that biological measurements are really heterogeneous. And so what I'm showing here is that on this LFP data that we had mice here, and what we're showing here is a TC embedding. For those of you that aren't familiar with TC embedding, it's a low-dimensional embedding that's only goal is to capture neighbor structure. So if you are close in this two-dimensional space, you're also close in that high-dimensional space. And other than that, the meaning of the components is arbitrary. So the components don't mean anything, but you preserve neighbors. And so if you were to actually do this, what we're doing here is we're coloring these mice so that each mouse is a separate color. And if you actually look at this data, if you take mouse 1 while it's being very active and mouse 1 while it's being very sedentary, that's much closer to each other than anything between mouse 1 and mouse 2. So a lot of the data is just individual variation. It's not actually the behavioral variation that you actually care about or the outcome variation that you actually care about. And so this is problematic, actually, in practice. And in fact, we went to say, well, if we're trying this convolutional neural network, so sure, there's a lot of heterogeneity, but this convolutional neural network is trying to find features that correlate with our outcomes, not find features that are correlating with individuals. And so, is this actually a problem in our convolutional neural network? Are we actually learning participant-specific features, or are we actually learning general trends that separate on these actual outcomes that we care about? And so to actually figure out if this was true, what we did is we took that same result we just trained, we took those extracted features, so we took that k-dimensional feature vector that came out of that first convolutional filter bank, and we said, well, this is what's being passed to our logistic regression. If this is what's being passed to our logistic regression, are these features actually very useful for predicting what individual we're looking at? So we took those same features, put it into just a standard classifier, and predicted subject ID. So this is saying, can we predict which subject we're looking at? And so this is our confusion matrix here, where we ran this with a cross-validation setup. And what's shocking here is if you look at this, a lot of these values are above 90%. And it turns out that these features, despite being trained to predict the outcomes, whether you were before or after treatment, they're actually much, much better to tell you which person you're looking at rather than which outcome you're looking at. Which actually, the first time I saw this, I would say really surprised me. Because I felt like we were trying to actually capture these outcomes. So the fact that we were actually not just capturing participant-specific features, but actually doing much better at capturing participant-specific features was actually quite surprising. And so there's this question of, and I don't have much time, so there's this question of, well, can we actually tell the network that it shouldn't do this, that it shouldn't capture participant-specific information? And so what you can do is there's an approach in the literature called domain adversarial neural networks. This is not their original use, but this is close to their original use. And so what you do is you have your nonlinear feature extraction, you get your low-dimensional extracted feature, and you have your logistic regression where you're trying to predict your label or outcome. That's a standard framework you do where you learn these things in an end-to-end framework. But what you do in this domain adversarial neural network is you actually take a classifier, and this classifier here is going to predict domain, and it's going to try to take your low-dimensional features and predict the domain as well as you possibly can. And so your logistic regression here, trying to predict the outcome as well as it possibly can. Classifier is trying to predict the domain as well as it possibly can. But where does
the person, right?
Person, yes. Domain is the person here, yes. And the key thing here is that you take your nonlinear feature extractor, it wants to do really well on this task, and it wants to confuse your domain classifier. It wants to say, we don't want to capture any classifier information. So you can say that this is an adversarial training because you're not actually doing this well. You want to learn features that don't do this. And so we can actually go through this. We remove participant-specific information in the feature space. So does this actually solve our problem? So because I'm asking this question, no, it does not. Right? If we actually go through and combine our sync-out approach with this domain adversarial neural network, we actually, not only do we not do better, we actually do slightly worse. And so this also, well, I say the first time I saw this was kind of confusing to me, but we started thinking about this a lot, and really the assumption is that all participants are identical in the feature space. And that's simply not a true assumption, right? People respond differently to treatment. People have different baselines. There are a lot of person-specific differences, right? So if you force them all to be identical in the feature space, pretty much the only way you can do that is by removing all information, right? Because all of it is somewhat predictive of participant in some way. And so we thought about this and we actually said, well, can we actually make a very less stringent assumption, which is to say, you don't have to be identical over all individuals, but you need to be at least similar to one other individual. So you need to have some similarity to at least someone else in the data set. So this allows you to have a lot more variability because you have this heterogeneity, but no one can be completely unique. And because I'm running low on time, I'm not going to go into this too much. I wanted to say it works by adversarial learning, which I really like this picture. If you ever look at adversarial learning, this is what you're going to see. It's also very useful for learning biomarkers in the clinical context, in addition to celebrity face generation and text word generation. So what actually happens here is that we actually get a side effect here, which is that, well, if we do this, we can actually draw a graph over which participants are most similar to one another, which is kind of cool. And so especially as our N increases, we're hoping that we can find cliques of groups of patients that are actually very similar to one another and actually explore this. With our current N, this is just really not feasible, so we can draw a nice graph, but it's hard to dig into how well this graph is doing. But then the question is, does this actually help us? If we enforce this type of structure, does this help us? And this was something called multiple domain matching network. And the answer is yes. This actually really does help. By enforcing the assumption that you should be not completely unique, but you don't need to be similar to everyone, actually turns out to be a really important feature, right? And this was something that actually gave us another pretty large gain. And so we're really excited about this because you know, this is really where we started. The first thing we did for them was this, and they were like, oh, this is great, there's information in there. And then we went from that to 60, and we're now at 68% accuracy. And I will say, we're pretty confident that it is improving. We did a Wilcoxon pair rank test, which is p equals 0.002. It's a little bit finicky on this test because we're running on a leave one out cross-validation, which has some assumptions that aren't truly satisfied there. So I will say take that p-value with a grain of salt. But we can look at this in a statistically somewhat rigorous point of view, and we think that it really is improving. But what's really exciting is that we think that these can be used to explore how the brain is actually changing. And so I'm going to skip this. But one of the things is, all right, so we got great results on n equals 25. So I have on my computer the stage 2 trial, which was a really funny thing because this is such a high-profile trial. They gave me all the EEG data, but I don't have any of the groups, so I have no idea who's in which group. And they will give that to me when the general public gets that information, and the general public gets the primary result of the clinical trial, and then I get to start on my secondary analysis. But that will hopefully be this summer. We're going from n equals 22 really to n equals 180. And we can add that initial cohort in if we wanted to, so we'll be at an n equals 200. But we also have a actual placebo and treatment setup, so we can actually look at things like treatment effect. And then I didn't really talk about the predictive medicine question. There's this question of can we actually predict how well individuals will respond to this treatment? We got above chance on that small data set, but we declined to publish it because the effect was just so weak and it was so variable and we had really bad outliers. And it was really these outliers that were really hurting us. So we're hopeful that with a much larger data set size, we'll be able to do the treatment effect a lot better from these neural signatures. And then the other thing that's really great is that Beyond NIH Autism Center of Excellence is going to run multiple clinical trials and studies on actually looking at the intersection between autism spectrum disorder and ADHD. So we can actually start trying to disentangle these two diseases better and use a lot of these techniques to try to understand which differentiates these different groups. So there's a lot of other things going on. One of the things that I think is really exciting is that we actually have people in computer vision that are actually putting apps on tablets so they can measure these individuals. Anybody that wants to download this app can, right? And there's computer vision approaches to automatically make sense of behavior while people are watching videos on tablets. We're also starting to spin up mobile EEG. So a lot of this stuff that we were doing where we were actually looking at device mapping across different things, we're really excited about this in the context of pushing this out into mobile so we can put this into mobile clinics or much more low-resourced areas, because if we have a universal mapping, we can just directly port all of our biomarker results between devices. And the other thing is that we're starting to get to the point where we can actually do digital phenotyping of things like autism spectrum disorder. So in a trial that we had, or a study that we had at Duke, if you use EEG alone on this similar age cohort, you can do it pretty well. If you use behavior alone, you can do it pretty well also. But if you start combining multiple sources of information, you can actually start doing digital phenotyping. And we think that we'll be able to do this with really low-cost resources that we could go and give to, say, every clinic in the country with minimal hardware investment. So we're really excited about that. There's a lot of other things going on. I clearly thought I was going to go faster than I actually did. But let me just conclude by saying that I'm really excited about actually trying to get more of these things into clinical trial data and trying to make sense of more types of data that we can actually use in order to get better biomarkers. And I think there's really strong evidence that we can actually start to make better use of a lot of these neural signals. And I am particularly very interested in trying to port a lot of these things that are being developed in animal studies into actual analysis in humans. So for any of you that are aware, the Brain Initiative was supposed to do both animal models and human clinical work. And in my opinion, the animal models got pushed a whole lot further. So a lot of the computational techniques were originally pushed in animals. It's really exciting to actually take what was developed there and actually try to push them into clinical use as well. Large-scale trials and clinical observation studies are currently underway, which is really great. Just real quick, the things I skipped over, one of the things is we're trying to build all this stuff so that we can actually use it to help improve clinical care and things like autism spectrum disorder. That's many years off. And so it's good to do things that you can actually do much quicker. So we've been working with neonatologists at Duke, where we're looking at detecting seizures in neonates in the clinic, and that's something that we'll be able to push into clinical practice much quicker. And we're actually doing really well on that problem as well, using all these same techniques. And we're actually testing out treatments of where we're actually using closed-loop brain stimulation in animal models. So we're actually figuring out ways of trying to actually build treatments in animals and try to do much better surveillance in actual clinical patients with all this stuff. And with that, there are a lot of people to acknowledge. These are my PhD students, somewhat. These two are really split. And then the Laboratory for Psychiatric Neuroengineering really helped me with all the animal work. The Duke Center for Autism and Brain Development clearly did all the clinical work. And funding for this came from a couple different sources. So thank you so much for your attention. Yes.
This was, go ahead, Elizabeth, please. You take it.
I want to know how you decided on 20 networks.
In the animal study? Yeah, so that was a little bit, what we did was we did a cross-validation on how well we could predict log likelihood of holdout mice. And so we didn't, in the initial studies, we didn't actually look at how well we could predict. And what was happening there is if you get more and more networks, you can actually explain more and more of the variance. But you kind of got that drop off past 20. And so it was really trying to look at a cross-validation technique of how much variance you were essentially explaining.
And an unrelated question is this chunking of EEG data. You gave us the number of samples you ended up with, but I'm wondering what the reasoning is behind that. How much do you take from one child EEG at baseline, say? Why not just take one chunk? But then you want more data, so then you've got more chunks. And you and I discussed once before the sort of correlation issue.
Yeah.
I mean, how do you decide how big the chunk, how many?
Yeah. So those are great questions. So in the EEG data I've shown you there, it's a 1 second chunk. And so the 1 second chunk is chosen because that's at the level where you can easily remove things like motion artifacts. So somebody actually goes through and the child like jerks their head like that. That data you literally can't use, but you can literally just like block out a second and you usually get rid of it. So people have found that a second is a good place where you can screen unusable data. So in response to the other question, which is, well, why not just use one window per observation per child? And you can do that, right? But we want to use all the available information. And so what I find very important is actually making sure that we're evaluating correct. And so that's why I'm very adamant about actually doing the leave one participant out. So you're working on a completely new participant, because if you're actually validating on Windows, if you validate on Windows, I can do something like 95% accuracy to tell you which group it is, right? And so if we were to only use one window on evaluation, then we would get stochastically the exact same accuracy results, right? Plus or minus some error, right? Because those are actually unbiased because we're just using lots of windows and averaging them. So you would have the exact same expected accuracy in this cross-validation scheme. And so the question is, can you get more information by doing this technique or not? And the answer appears to be yes, based on a lot of empirical studies, but I don't think there's really great theoretical reasons for it. Yeah.
So when you're able to predict patient identity so well, and then do worse when you apply the domain adversarial network, I just told me you were overfitting the model in the first case.
Oh yeah.
So how did when you did your next iteration of that, could you still predict patient identity?
Yeah, so I don't have it in here. So basically what happens is, kind of this graph structure. You see that you actually have a lot of links to similar patients. So if you look at what happens to this, really what happens is those clicks start to show up a little bit, where you can still tell which patient you're looking at pretty well, but within those little clicks you do much worse telling apart from them. But you can tell almost certainly which click that particular sample is from.
Also, and in a previous slide, it looked like you were predicting, I mean, that's not understand what the abscissa or the organ was, but you were predicting better at 12 months out from 6 months out.
So that was just a feature score. So this feature score started at baseline and then 6 months and then went up to 12 months. And so there's a comment on that, because one of the things you might think about doing is treating this as an ordinal variable rather than treating this as a 3-class. And so actually, based on the conversation I had with the clinical experts there, they really wanted this as a 3-class classification, because a lot of individuals don't start showing responses till a year out. And so there could be very nonlinear response curves.
What was the clinical setting here? Can you just remind me? These kids were getting, patients were having EEG.
They had EEG, and I didn't go into too much of the behavioral paradigm, but they were basically watching videos. These videos were designed to actually look at social and non-social situations.
Okay, but you expect a fall off, right? When instead it has sort of a U shape, right? I mean, that's just unexpected, right?
That's what I expected as well. And they, when I was talking to the clinical experts, they said that that's actually not uncommon on a lot of these treatments, is that you get some bounce back after a while. So I actually expected exactly what you were saying, which is for it to go like this. But when I talked to, or vice versa, right, if it goes up to kind of flatline, or continue growing. And when I was talking to them, they kind of see a lot of these nonlinear trends. So this was actually unsurprising to the clinical experts, and I don't think that that's very well understood.
Okay.
Which I realize is a very unsatisfying answer.
No, that's fine. I guess one last quick question I have to ask is you did all this sort of wavelet convolutional neural network. But did you ever try this as a regular convolutional neural network?
Yeah. So it doesn't do very well. It overfits terribly.
Really? I mean, with the same architecture, but just leave out all the wavelet stuff. Because you can still, you take the filters and you can interpret those just fine.
So
right?
You could argue that you can, but the thing is, if you do that, what really happens there is you have so many more parameters in your convolutional neural network that you basically go, so this network only has a few hundred parameters. If you put on a standard convolutional neural network, you get way more. And so it overfits pretty badly if you do that and you don't constrain these filters. And that's largely going to be down to sample size, right? If you kind of get this really large sample size, like if you get in the n of thousands, you would actually expect that your standard convolutional neural network would do as well, if not better, because it could actually capture more variation. But when you're looking at these very limited sample sizes, you really can't go and apply those things out of the box.
Okay. And there's other ways to regularize it.
Yeah, so...
Understood.
Yeah.
I have more questions, but let's do it offline.
Okay.
Thank you so much, David.
Yep.